Hello, everyone. It's a pleasure to be here, even if virtually. And I'm happy to tell you about my work on the quest for deep knowledge, decoding the human genome with deep learning models. Perhaps the most fundamental question of biomedicine and biology is the mapping of phenotypes to genomes. True molecular understanding of human disease requires us to fully map specific coding and non-coding variants to their biochemical and phenotypic effects through the complex cell-type-specific molecular circuits. To map the connection between genome and phenotype, we thus need to predict specific functional impacts of variants through mapping of cellular circuits to precisely identify their phenotypic outcomes. In this talk, I hope to demonstrate to you that this task can only be accomplished through tight integration across human studies, tissue culture, and model organism experiments, and sophisticated computational approaches that are able to interpret the genome in an unbiased and data-driven way, as well as map molecular circuits that these genomic changes are affecting in the variety of different cells and organs in our bodies. So before we go much further, I really want to emphasize to you that when we talk about genomes and variants, of course, there is not just the coding space of variants, but also the vast over 98% of the genome that is non-coding and of course affects the various regulatory roles in the human cells. And what we really need to do is to go beyond the approach of looking statistically at a specific non-coding variant present in a given subset of the population and trying to associate it with phenotypes just that way. That's a very powerful way, but it is not sufficient because many of those variants are very rare. We're not going to be able to find them in the same genomic context. They will have different outcomes, and often multiple non-coding variants are actually going to be needed to detect a specific phenotypic effect. So what we really need is a similar sort of code that we see in the coding variant space, where we have our genetic code that's familiar to us from every textbook in genetics or molecular biology that students learn in their first class. We kind of need a similar effect regulatory code where we're able to map every possible non-coding variant, be it common, rare, or perhaps even never seen before in sufficient numbers to be able to make any observations in human populations. We need to be able to map that variant to specific biochemical effects on binding of transcription factors, epigenetic outcomes on histone marks, as well as post-transcriptional effects. We find that actually deep learning can accurately predict impact of any variant at the transcriptional and post-transcriptional level. And of course, our DeepC approach has come out in 2015, and there's been a huge blossoming of this field, and there's a number of methods of different types of deep learners, both at the transcriptional and post-transcriptional level. And at the post-transcriptional level, we're specifically interested in a variety of different effects mediated by RNA binding proteins, be that splicing or RNA stability or transport. And that's our seq-weaver method that I will also discuss today. Critically, these models do not learn on any mutational data when they're training. So that means that they can predict the impact of any variant, even rare or never before seen variants, with single nucleotide resolution. Just to give you a little bit of an insight into how these models work, since they're published, I'm not going to go into huge detail on the deep learners themselves, is that we actually use at the base level the hierarchical structure to capture the context information of the sequence. Of course, the multi-layer nonlinear transformation is allowing us to really capture the interaction of sequence features, which we know biologically is quite important. And finally, at the output layer, we really can leverage multitask prediction so that we can leverage cell types where there is less data, where there's more data, to help us gain information about predictions for related cell types where there's a lot more data. And of course, that all reflected in this hierarchical structure where the convolution layer is really where we're scanning for motifs, then spatially scaling this in the pooling layer, and finally really capturing the higher order sequence features and interactions in the convolution layer. At the time, we and others did not know whether this was possible to do, but it turns out that now we're in the field quite familiar with these models. And while they are not trained on any mutational data, once you have a trained model, such as, for example, DeepC for transcriptional variant impact prediction, you can apply it to any variant. So in this case, I'm just showing a hypothetical variant on chromosome 7, a change from A to C, and the model can predict impact on thousands of specific features, so specific histone marks, DNase accessibility, as well as transcription factors. And then what might be one of those predictions is, for example, a significant effect on a transcription factor binding where, let's say, your given transcription factor of interest can bind tightly to the A allele and no longer binds or binds very weakly to the C allele. And this perhaps could be used to explain the differences in phenotype that are associated with this particular variant. So these models are actually quite accurate. We've evaluated them extensively, both computationally as well as on external experimental data sets and on our follow-up experiments. I will just show you a number of papers that have used these models on everything from AQTL prioritization to integrating them in polygenic disease risk assessment to specific phenotypes like blood pressure, etc. So when applying these models to full genome sequencing data, here I'm going to show you an example where we're going to apply it to, in the context of autism, to over 7,000 whole genomes in the Simon Simplex collection. We're able, for the first time, to actually demonstrate significant burdens of non-coding regulatory de novo mutations. And this was actually true for any human disease, but here, of course, it's shown for autism. What we actually did is we identified this burden both at the transcriptional and post-transcriptional level, as you can see here, even though this burden was not detectable at the level of mutation count, as you can see in the red and gray bar under the de novo mutations error. So the mutation count for the probands and siblings, unaffected siblings, was actually nearly equal on average, whereas there was a significant difference in how bad by these models were predicting these mutations of an effect these mutations to have. So essentially, the regulatory variants that ASD kids had were worse in their impact than those in the unaffected siblings, even though they did not have significantly more variants by count alone. This turns out to not be unique to autism. We now have another paper published in Nature Genetics in collaboration with Bruce Gelb's lab from Mount Sinai and others looking at congenital heart disease, and we're actively working on these questions in other diseases as well. We have actually experimentally shown that a number of variants that we prioritize with high predicted impact do in fact drive expression differentially in luciferase reporter assay. These, of course, are very interesting candidates for future studies, and what I find actually also very interesting is that there's approximately equal number of variants here that cause an increase in expression of the gene for the affected ASD individual versus a decrease. So this is a single letter change, a single nucleotide change between the proband and the sibling causes this change, significant change of expression in the luciferase reporter assay. Now, of course, this is not just at the transcriptional level. As I mentioned, we actually had a separate model, SeqWeaver, that was built specifically for looking at post-transcriptional effects. And we call these RRD regulatory effects. And I just wanted to show you one of those variants. And I'm showing this to you actually in a human-based visualization where you can access all of our predictions and you can browse them and get them in detail, look at the specific predictions based on different features and cell types, etc. And so this particular one is a prediction of a regulatory variant that is affecting SMEC1. So SMEC1 is an important protein. It's the regulatory subunit of a serine threonine kinase. And it's actually we could tell that it was not just a general RNA processing effect, but splicing, because the top RNA binding proteins for this variant that our model was predicting to be affecting are all splicing factors. Now, SMEC1 has never been associated with autism specifically in any way, but is an important protein that's highly conserved. And in fact, when we actually look at this experimentally, indeed, RRD mutations can disrupt splicing. So we do find an ASD proband-specific reduction in the long isoform of SMEC1. And this was actually done in the lab of our close collaborator, Bob Darnell, who is co-senior author of this paper. And you can see that this is a significant reduction in ASD probands. And in fact, when we look at the RRD mutations in general, we actually find a significant association with phenotype, in that low proband IQ is significantly associated with higher dysregulation at this RNA processing level in the kids with ASD. So the kids with ASD who have lower IQ also have significantly higher dysregulation of their RNA metabolism identified by our models. So then that really raised the question for us of, is this a general pattern? Is RNA binding protein target site dysregulation actually causal in psychiatric disorders in general? And of course, RNA binding proteins are incredibly important. They regulate all steps on the RNA life cycle, and in fact, have been associated with a number of psychiatric disorders before, but from the perspective of mutations in the RNA binding proteins themselves, right? So what we are talking about is the target site dysregulation. So this picture on the right. And that's critical because while RNA binding proteins themselves, their mutations in them would be targeting that one particular gene or a couple of those genes. Over 90% of the genome, of course, are these non-coding regions where a mutation in that area could actually prevent a perfectly functional RNA binding protein from binding. And could that actually be something that leads to psychiatric disorders? So that's what we're asking here. And just, I wanted to show you how we look at this. So what is the framework we're applying? Of course, we're going to use seqWeaver, the same model that I've talked about before, for being able to predict the effect of any of the variants on binding of a number of RNA binding proteins. And this model, by the way, is trained on in vivo data and allows us to look at a number, over 200 different RNA binding proteins, and spans effects beyond splicing, such as stability and transport. So we applied stratified linkage disequilibrium, LD score regression model, as the statistical framework for partitioning disease heritability into various functional annotations, while directly modeling the extensive LD structure between SNPs. By combining this LD score regression with our deep learning framework, we can now directly estimate the contribution of RBP dysregulation to psychiatric disease. So we're going to use GWAS studies from the Psychiatric Disorders Consortium to be able to assess this. And I want you to be familiar with these graphs because that's what I'm going to show you. So we're going to look at these graphs where on the x-axis we're looking at the psychiatric disorder risk increasing towards the right, and on the y-axis we're looking at RBP dysregulation, again increasing towards the top. So the lower line, if we see our data following along that sort of line, then that looks more like there's no evidence of association in that particular GWAS study. Whereas if we actually see that higher line where the higher psychiatric disorder risk is associated with higher level of RBP dysregulation from the stratified linkage disequilibrium framework, then that implies causal association and causality to disease risk of RNA binding protein dysregulation. And here's what we see. So here's the most well-powered disease, and that's schizophrenia. And you can see that we find a very strong evidence for association between the RBP dysregulation and disease risk. And that's actually true across a number of diseases. So for ADHD, ASD, bipolar disorder, major depression, and schizophrenia, which are all the studies that we've looked at that had sufficient data, we find that there is association. I would not look too much into the size of these right now because Of course, there is also the power of the original GWAS that we're studying. So I don't think we can conclude anything about the fact whether there is more of this effect in ASD versus schizophrenia or less of this effect in ASD versus schizophrenia based on that. But what we do see is in every one of these psychiatric disorders, we see this association. And of course, many of these RBPs also have large number of coding mutations associated with neuropsychiatric phenotypes. For example, EF2UD2, so that really implies that there is a convergence of effect both on the non-coding and the coding end that's really likely to cause psychiatric disorders. You can actually now use our website on human base, so hb.flatironinstitute.org, and you can look up all of these data. So you can put in a protein you're interested in. So for example, in this case, I'm putting in CACNO2D3, and then see the results in this heat map. You can actually mouse over and get a lot more detail. And actually this one particular, we did actually, so this is a transcriptional effect outcome, not post-transcriptional, but we did test this particular one with an in vivo expression reporter, and it turns out that particular variant that we predicted to have an effect indeed does control expression and change it in a significant way. Now, of course, these variants aren't acting by themselves, right? These proteins are part of the complex molecular circuits that are highly tissue and cell type specific, and it's really important to be able to understand how various proteins, once they're expressed, how they're interacting in these circuits. And so we're also very interested in mapping these cellular circuits with tissue-specific functional maps. So the goal here is really to create maps of how various proteins interact in different cell types and tissues in the most data-driven way possible by using the large amount of functional genomic data, expression, physical interaction, etc., that gives us some view at how this is happening in a functional podocyte in the kidney or in a particular neuron in the brain. So to do this, we now can take advantage of thousands of diverse genomic data sets that we have available in human and combine it with the tissue-specific knowledge. The tissue-specific knowledge could be coming from human or from mouse. So for example, I will show you a little bit of a study where we actually use mouse neuronal specific signatures that we map to human to identify which genes are expressed in very specific types of neurons to be able to actually understand neuronal cell-specific vulnerability in neurodegenerative disease, in that case, Alzheimer's. And we can use these connections as well as known pathways in human to be able to essentially use it as a hook in our Bayesian data integration. So we can use these gold standard analysis for semi-supervised regularization in Bayesian classifier to be able to assess the evidence of how relevant and accurate each data set of those thousands of diverse genomic data sets is to the particular question we're studying. So let's say to a particular neuronal subtype in the brain. And then we can integrate these huge numbers of data weighted by their relevance to that particular neuron to then assess the probability of tissue or cell type specific functional relationship between two genes, and do that for every pair of genes in the genome so that we can get a functional genomic map that is essentially a summary, a probabilistic summary of all of the data available that is done in a way specific to that cell type or tissue of interest. So let me show you that these actually work first. They're accurate, and they're actually able to effectively extract tissue or cell type specific signal even from highly heterogeneous collections that might not even have any data that's coming from that specific sub-cell type or little data from that particular tissue. So in this case, we're looking specifically at IL-1 beta, which is of course an important cytokine. And we were working with collaborators who were very interested specifically in its role in cardiovascular disease. So specifically, they're of course interested in the blood vessel cell type. And we are looking here at the top 20 interactors predicted by our integrated network, specifically the blood vessel network, to be interacting with IL-1 beta. So this is, again, the interface that I'm showing it to you is directly human-based that I've referenced before. And it turns out that when our collaborators actually evaluated this and basically looked at which genes changed their transcriptional levels, their expression levels, upon activation and treatment with IL-1 beta, it turns out that the very first time point, 18 out of 20 of those genes were significantly affected, and it turns out that actually the other two are just not transcriptionally regulated, but also correct. But even if we just looked at 18 out of 20, that is a highly significant p-value. And what's also important is that accuracy of the blood vessel network for this is significantly higher than other cardiovascular networks and much, much higher than global network or those from unrelated tissues. So this was the most accurate network than any of the other cell type or tissue specific or global networks as well. We can actually use these networks as well to understand the network architecture of disease. So here I'm actually looking at AD, GWAS catalog genes, and I'm looking at the hippocampus network, and you can see immediately that the clusters that we are identifying are actually clusters that correspond to key processes known to be involved in Alzheimer's disease, such as memory, learning, amyloid beta formation, etc. Oops, sorry. So we can actually, in the human-based interface, then zoom into a specific one of these proteins and be able to look at those genes. So this is 5 genes from the M1, from that amyloid beta formation module in the hippocampus network, and we're able to look at them in more detail, and you can zoom into particular interactions. So for example, here we're looking at the interaction between MAPT and CA-MK2B, and you can see that the most evidence used for this prediction is co-expression, but there's also data from GSAA perturbation. So you can actually go in and figure out exactly why the network made the prediction and what evidence was used, and actually go in all the way through to specific papers. So now that I showed you how we make the networks in the interface, I wanted to go back to that study that I briefly mentioned about untangling the basis of neuronal vulnerability in Alzheimer's disease using these networks. And this is in close collaboration with Paul Greengard's, late Paul Greengard's lab at Rockefeller, and the really incredible experimentalists that we work with there, Jean-Pierre Rosery. So cell type and tissue specificity is a key aspect of many complex human data sets. And in fact, it can be critical in understanding the molecular basis of such disorders. Alzheimer's is clearly one such case where we need to peer into very specific neuronal subtypes that are specifically vulnerable to disease to identify vulnerability of specific processes. We were able to use backtrap data from mice to measure actively transcribed RNA in specific neuronal cell types in mouse. And then we use it to provide cell type specificity to generate human networks for these cell types from very large compendia of non-cell type specific human data. And I've showed you this on the computational approach slide. So hopefully this actually is now making sense in the context of this specific disease. Then we can actually use machine learning of these vulnerable networks to prioritize AD-associated genes and then to map them onto specific functional modules. So I've mentioned this to you, and I've showed you how we do this again. Once we build this network, right, using the mouse data from neuron-specific signatures that we map to human, we can now use these networks to identify candidate genes that are key to AD-associated vulnerability. What we do is we take the top hits from a quantitative genetics study, so in this case, Alzheimer's disease GWAS, and look at where the nominally significant genes versus the non-significant genes fall in the network, essentially using the network neighborhood as features for a supervised machine learning problem. We developed this NetWAS 2.0 approach specifically for this problem in Alzheimer's, where it doesn't just do this, but it probabilistically subsamples non-significant negative genes to take advantage of the GWAS confidence scores, which makes this method even more powerful and robust than our original NetWAS approach. So here we use the original Alzheimer's disease GWAS and the specific vulnerable neuron network to be able to prioritize all the genes in the genome by their association in Alzheimer's disease, specifically based on this neuron-specific vulnerability. We find that these prioritized genes are indeed enriched for Alzheimer's disease-related signals. So here I'm showing you the enrichment in two sets of processes that are both considered hallmarks of the disease. And we see strong enrichment in both, significantly more so than the original GWAS study shown here. So placing these genes, prioritized genes on a schematic neuron, we actually identified the vulnerability-specific functional module functioning neurotransmitter release and located in the axon of these vulnerable neurons. This allowed us to make specific hypotheses about a mechanism that may contribute to this selective neuronal vulnerability in AD. And two key players that we identified are tau, one of the major players that we do know about in Alzheimer's disease, and interestingly, alpha-synuclein, a gene that has been implicated in other neurological diseases. And looking at what is connecting both of these genes, we found PTB, a splicing regulator. And in fact, follow-up experimental results actually preliminarily show that it does indeed regulate tau and alpha-synuclein in this neuron type, and our collaborators are following up on this deeper in both model organisms and human cells. So I want to thank you all for listening to this talk, and I hope that I've given you a sense of how we can actually use these advanced deep learning models as well as Bayesian approaches to really develop predictive models that eventually, for now, can interpret the genome and decode the genome as well as map biological processes. But in the future, we hope really can allow us to model complex molecular, cellular, and organismal systems at complementary levels of resolution, and really to be able to integrate molecular biology with whole organism physiology. And thank you very much. I really appreciate the opportunity to speak here.